Hello all, my name is Samira Al-Masri, and today I will be presenting to you my final presentation for Systems Bio over an article titled B-Cell Failure as a Complication of Diabetes. So this article discussed some of the complications with diabetes in relevance to B-cell failure. The authors of this article were K.J. Chang Chen, R. Muller, and E. Bernal Mizarchi. This article discusses the complexity of type 2 diabetes. So it discusses in particular beta cells in the pancreas. So normally, pancreatic beta cells help control the blood sugar levels to return to bodily homeostasis. So when we eat too much sugar or even right after we ingest a meal, beta cells help regulate our blood sugar. Now for people with diabetes, they have an insufficient insulin release, so they don't often release enough insulin to allow those levels to return to homeostasis and optimal body function. So this would be an example of a negative feedback loop, which is a self-regulating system within the human body to regulate hormones, blood sugars, nutrients, and so forth. And the release of insulin is done in order to reduce glucose levels within the bloodstream to allow for a balanced homeostatic condition. Now, with diabetes, this is seen when there's a complication within that negative feedback loop because there's an insufficient amount of insulin that is being released, and often they have higher blood sugar levels than what is ideally healthy for the individual themselves. Now, why did I choose to study this? I had an interest in negative feedback loops within the human body, and specifically complications within them. So complications in negative feedback systems contribute to various diseases. So for example, hormonal imbalances like hypothyroidism or diabetes results from disruptions in negative feedback loops that control hormone levels. And additionally, autoimmune diseases can occur when the immune system fails to regulate itself properly. And that happens within the negative feedback loop. And that can be where the body even attacks its own tissues. So these are just a few examples of disruptions within the negative feedback mechanisms that affect various physiological processes. So with diabetes in particular, according to the World Health Organization, it is projected to rise to affecting 643 million people by the year of 2030. So my motivation was to understand beta cells' role in diabetes, for the relevance it has to our future, and my interest in complications within negative feedback loops. In summary, this article discusses the complexity of type 2 diabetes, focusing on beta cell failure in the context of insulin resistance. It emphasizes the adaptations of pancreatic beta cells to insulin resistance by increasing the mass and function in early stages of the disease. So this also is followed by the negative impact of persistent nutrient excess on the beta cell function. And in addition, this paper reviews the regulation of beta cells mass in normal conditions, mechanisms of beta cell expansion failure, and beta cell failure in diabetes itself. It also discusses the contribution of neogenesis to the maintenance of beta cell masses and failures within that mechanism as well as the role of cell cycle proteins in B cell proliferation and the modulation of B cell masses within adults specifically. Lastly, it also explores the effects of glucagon like peptide 1 and glucose-dependent insulinotropic polypeptides on B cell proliferation and apoptosis. It delves into insulin receptor signaling, the role of insulin like growth factors, for example, IGF-1 and IGF-2, and their receptors in regulation of B cell proliferation. So here we have a beta cell failure diagram. During instances of glucotoxicity or lipotoxicity, which lead to the production of ROS, in which we can see right below the oxidation, it activates J and K, which is also following that arrow from ROS. And J and K activity then leads to decrease in the IRS, which is the insulin release substrate signaling. In these processes, it induces stress within the ER. The ER stress response and its effectors are then activated in response to the misfolding of proteins performed by this process in order to try and protect the beta cells from apoptosis. But despite this, activation of these processes under long-term conditions with elevations of FFA and glucose, it can lead to beta cell dysfunction, and then, in this case, ultimately apoptosis. One interesting finding noted in this article is that inhibition of the IRS signaling seems to be a common pathway induced by the majority of mechanisms described in beta cell failure. An additional event is the increase in mTOR signaling by nutrient access glucose. And furthermore, going back to one of the big reasons why I wanted to do a project like this involving negative feedback, was this results in negative feedback inhibition on IRS1 and possibly IRS2 depending on the conditions. The decrease in IRS signaling induces GSK3 beta and FOXO1 function, and this activation of these molecules reduces PDX1 levels and increases the levels of cell cycle inhibitor P27, as we see down right above beta cell dysfunction, which then leads to apoptosis. Here we have the initial Cytoscape upload of the data and the identification of the primary nodes. Next, we have the data set that I got from Index. And then after, here is the identification of the nodes. So one of the main nodes in beta cell apoptosis is IRS. And IRS stands for insulin receptor substrate, and there's 1 and 2. In that, there are 77 amino acids, and the key role of this substrate is to mediate the control of various cellular processes by insulin and other cytokines. So here I also put the entry name, and then in this figure, although it's a little hard to see, we can see different connections to insulin, TNF, IGF-1R, the MAP protein, mTOR, which is right by the little box right here, and the INSR, which is the insulin receptor. Next, using Signor, we identified TSC1, which stands for tuberose sclerosis 1 protein. And it is a protein. It's in the complex with TSC2, and its key role is to inhibit the nutrient-mediated or growth factor of proliferation by negatively regulating mTORC1 signaling. So this protein also downregulates CDK, ERK1 and 2, and MAPK1. It then upregulates AMPK and FOXO3. And here we can see the different interactions of upregulation and downregulation with the diagram to the right. So next we'll discuss the summary of the findings themselves of the article. Overall, the article had discussions of mechanisms of beta cell failure, and the key factors included the generation of reactive oxygen species, ER stress, alterations in beta cell metabolism, decreased IRS signaling, and induction of pro-apoptotic signals. And in the findings, we also see that the results of a lot of these processes are apoptosis. In addition, it discusses J and K activity, which leads to a decrease in the IRS signaling, and also an increase in mTOR signaling by nutrient access results in negative feedback inhibition on the IRS1 and possibly IRS2 substrates. And lastly, a decrease in IRS signaling induces GSK3 beta and FOXO1, which then reduces PDX1 levels. So the projected nodes that would be targets for therapy after reading the article and delving into some of the networking and interconnectedness between the genes would be the ones listed here. Primarily, in my opinion, mTOR and TSC 1 and 2, along with IRS 1 and 2, would be ideal targets for therapy. Next, we have pathway regulators as targets for therapy, and after looking at the different networks and such, we have a list here. I would more specifically emphasize cyclin D2, mTOR, and FOXO1 as the targets for therapy. Now, here is just a slide to show that the author is built on previous research with beta cells. And lastly, why is this appropriate for systems biology? So systems biology can be used to interrogate complex diseases, looking at multiple different nodes in relations to networks. And pancreatic beta cells control blood sugar levels to return to bodily homeostasis through negative feedback loop. And with this, diabetes results in an insufficient insulin release, and this disease in particular is very complex with many different nodes and such, and even things that can go wrong with different substrates, proteins, and so forth. So I thought it would be very, very interesting to look at this relationship between all the factors. This complex network can be broken down with the learning outcomes of systems biology to look at the different connections and interactions among the host genes and products and their interconnected pathways. And then in addition, the basis of network and graph theory can be used to analyze these biological connections and develop a better understanding of the disease and possible target therapies. So thank you guys very, very much for listening to this presentation, and I appreciate your time.